be okay. All right. Like Donna mentioned, I'm pretty much grown up on this campus, UCSD. And today what I'm going to talk to you about is an idea. An idea that literally it didn't start that way. It started with we finding one molecule, then a family of molecules. Started trained as a physician, tried to look where would it mostly impact which disease process. And a lot of my lab works on cancer. Show of hands, how many of you either have had cancer or have had near and dear ones suffering with cancer? Thank you. Me too. The topic today is Herculean task of killing cancer cells, and it is Herculean, but most of it, what is being done in the clinic, is chopping one head at a time of this monster. And we all know, as the proverbial thing, that one head chopped off, two grows right there. So it's kind of futile. I'm going to walk you through what we have found. So I am hoping all of us here who have seen somebody suffering from this disease. It's costly because it's basically a bunch of cells gone rogue. It continues to proliferate, continues to spread, continues to evade our immune system and keep spreading until we're consumed. Until, in few situations, it could be picked up early, but that's it. How many of you watched the 3 episodes of Cancer, the Great Masquerader on KPBS? The 3 very, very well done episodes. So, you know, the topmost people, the researchers, all waiting for the Nobel Prizes, they couldn't look into the camera, shifting eyes, admitted 44 years since President Nixon declaring war on cancer, we have been pretty much running on the treadmill at one spot when it comes to treating solid cancers. Not hematologic, but solid cancers, colon, breast, lung, you name it. So part of the reason is there are some facts that's common to any cancer. So I'm going to touch upon those quickly. Fact 1, none of us would be worried about it if it weren't a metastatic disease that is spreading. So if cancer spreads, it kills. If it were to stay where it is, every surgeon can have as much possible amount of tissue cut out, the tumors out, we would all be okay. So that's a fact. Second fact, most cancers, actually all cancers, are driven by just aberrant signals. Signals that make it go rogue, that we talked about. Normally, every cell of our body has the checks and balances to divide only when needed, to move only when needed, to die if they are going to come off of the attachment, they should die. That's the programming we all should have. Except cancer cells have acquired certain abilities that will make them go through self-sufficiency, evade the apoptosis pathways, which is the cell death pathways, keep making new blood vessels, angiogenesis, keep replicating limitlessly, invade, metastasize, so on and so forth. That's a fact. It's a, sorry, that's my 5-year-old pickup alarm. I apologize. So, fact number 3. Most of the time, the reason normalcy exists is because we have those checks and balances. If there are signals being produced, they are from every possible receptor on the cell surface. For example, look at these. There are some famous ones, HER2, EGF receptor, all of them are drug targets currently being used in cancer clinic. But normal cells would take those receptors, if they're signaling, they'll be degraded inside the cell in our big garbage bag, which is the lysosome. They're organelles that'll chew it up and spit it out. That's normal, except cancer cells have a way of kind of letting that signal go out of control. Fact number 4, here is a slide that I'll walk you through it. All these different drug names, they are currently being used to treat cancers. Look at the progression-free survival that we can achieve with any of them. They fall around this little big, the big red dot area, most of them. Here is the response rate percentage. So about 60% of the time, we get a 10 to 20 months of progression-free survival, which means we treat and the disease is not there. But most of the time, it comes back right around this time, and therefore we call it disease has progressed. Now, this is our goal. Look at this. This is discovery of Gleevec, and that is a liquid cancer. That's hematologic cancer. That's considered cured. That was one of the miracles of modern medicine. But what has happened is that hope for miracle pill, the discovery of Gleevec, the hope for miracle pill never, ever came through in solid tumors. Just didn't. And there's one good reason why. Because solid tumors, every tumor cell could be different from the other one right next to it. So if we think that my tumor is different from another person's tumor, that's not enough. Even within my tumor, every single tumor cell has an ability to actually be slightly different, ever so slightly different, so that the same drug may or may not work. And so we'll select the population of cells that have escaped the drug that is being given right now. So having said that, this is also another fact. Numerous clinical trials have shown what are we accomplishing with a chemotherapy. We are simply going ahead and selecting new cells that have made sure that they could develop resistance to the drug that I'm currently treating the patient with. So we have been blindsided time and again by the unknown number of pathways and mutations that can feed the tumor as soon as we block one pathway, one receptor, one single side of signal transduction. So I tell my students, it's kind of grabbing a wet bar of soap in shower. It keeps slipping away. So here is how I teach signal transduction in cells. I lie all the time. Because to those undergraduate students, graduate students, I tell them signaling is like this. It's like receiving a phone call. Signal in, signal out. For a cell phone, that'll be a radio frequency wave, and then there's a voice coming out. And that's exactly how it happens in a cell. Signal in, and then signal out. That's not how it is. This is more like how it is. It's a mess. It's a mess because turns out every receptor on the cell surface could signal through the same group of molecules to go ahead and affect gene transcription. Very soon, when the same undergraduate student will become a PhD student, they'll realize it's kind of like this. So all that is oversimplification. So that brings me to my next slide is I don't own any Apple products. I don't buy them. I don't sell them. But I buy what this man says. And of course, most of you will agree. He's the one who brought up the distorted reality field. He will put together a vision completely impossible to accomplish, and then lo and behold, everybody will make sure they will accomplish. So, simple can be harder than complex. But if we took some time to simplify it a little bit, maybe we can move mountains. And here is where I come to looking at the internet. There is something very, very, very same between internet and cell signaling. So it turns out in the year 2000, 4 Silicon Valley companies got together, IBM included, and they looked at how people surf webs. Turns out there are several millions of pages from which you can surf through a central core of few pages that can, again, lead you to several million pages on the other side. It's unidirectional. Notice the arrow can go only one direction to the other, never this way. And it turns out that there's a lot of flexibility here and a lot of flexibility there. But here, there's very little flexibility. There is rigidity. There is robustness. There's precision, whereas these sides are not so. Turns out, this is the fundamental of information flow. And turns out, biology also uses the same principle to flow information within every cell, within every minute of the day. So, biology has evolved the architecture of bow tie architecture of information flow in almost everything you can imagine. In all those systems, there's diverse input of signal. There will be universal, invariable, limited number of robust carriers in the middle, and then there will be fan out of diverse outputs. Rarely, and I mean it, rarely, we may be able to have a universal control if this region can be controlled by an on and off switch. So just to let you know, this is a way for information flow to be layered. This is a way for information flow to be controlled, layering of control. Don't worry if this doesn't make sense right now, but let me share with you how many things use a bow tie method of communication. Here are just some examples, okay? Look at it. I started with the internet, but signaling networks is what I'm going to focus on today. But metabolism, developmental regulatory gene network, our immune system, our visual system, our neural networks, our human language and writing, everything uses that fundamental way of making information flow. So that brought us to looking at our favorite system that we were trying to solve, how to think simplistically about signaling networks. Look at it. Multiple receptors can signal through a few key intermediates. These are cyclic AMP or calcium. And then they can regulate the expression of multiple genes. So right away we knew that this is where the key is. That is the point of information flow. What is known is small changes in that region can make the biggest impact downstream. It is also known that small changes in that region has the ability to capture the biggest amount of information that is coming in to make that biggest amount of impact downstream. Okay? So that is just the network of information flow. Now, before I go to why on earth did we get into this? Because I know I was trained in physics and math, but I never knew I would use it this much until now. So here is what that small molecule cyclic AMP is. If you wondered, what is cyclic AMP? Well, to tell you, to a molecular student who studies structure, this is it. It's a small, very tiny molecule. It can diffuse inside a cell within few seconds. That's why I call it a bomb. And what it is it is kind of a bomb. Each G protein that will get activated by a receptor can then activate 10 adenyl cyclase molecules. That'll make 5,000 cyclic AMP molecules. That is going to eventually lead to 2.5 million protein phosphorylations. So this is a domino effect, and the largest step of amplification is production of this cyclic AMP molecule, this tiny little molecule. It is communicating between the outside of a cell, the receptor that is here, and inside of a cell where all the phosphorylated proteins would act on gene targets. So what is cyclic AMP? Continuing with it, here is the best part. Turns out, to produce a cyclic AMP, you're going to eat up ATP. ATP is the energy currency of a cell. Without ATP, no cell will be alive. It is the way how a cell makes its purchases, his credits, his balances. So think of it, a cyclic AMP produced is going to use up energy from a cell, the ATP source, make a cyclic AMP molecule. It'll be chewed up by, of course, some other enzymes to make AMP, adenosine monophosphate. But enough said. What is it about cancer? Turns out, cyclic AMP protects against cancer. Having high levels of cyclic AMP is actually good. No matter how you accomplish it, you could accomplish it by making more of it, or you can accomplish it by preventing the degradation, or you can accomplish it by actually having activation of receptors that are going to trigger more of it being produced. What is known is it's sufficient to inhibit proliferation, invasion, basically metastasis and growth of cancer cells, of a variety of cancer cells, not just one, across the board. So makes sense. If you have something at the level of that hourglass, of the information flow, something that is invariable, something that is rigid, something that will capture everything above and then relate to everything down, it should work across all cancers. Made sense. But this is all not our work, but this is all evidence, 40 years of work. And then I'm going to ask you the question, how do we know how cyclic AMP works to inhibit cancer? Nobody knows, but one of the theories is that it robs the cell, the tumor cell, of whatever little ATP it has. Cancer cells have the least amount of ATP in the entire body. All the normal cells have plenty of it. The cancer cells have chronically very, very low ATP. It has been shown that if you simply take away those few ATP molecules, you can actually stop that cancer. Or if you feed it tremendous amount of ATP, you can make it grow more aggressive, into more aggressive form. So this could be one way, but I'm just leaving it there. There's no hardcore evidence supporting that. Now, here is where we are. How can we control it? How can we, or how do cancer cells control the cyclic AMP? How do they know how to get it low? So here I have put the cyclic AMP right there at the bottom, in the node of this bow tie. And I'm hoping through the next few slides I'll tell you we have stumbled upon, not gone looking for it, stumbled upon such a nice target or a bunch of targets. And here is how cyclic AMP is generated in cells. I will take a few minutes to explain just this simple part that I've put out, because even though it is simple, 8 times it has been picked for Nobel Prizes. Just that little bit on that board. 8 times 2 to 3 people each time. So that makes it almost 16 to 20 Nobel laureates, just that much. So cyclic AMP is produced by these G protein coupled receptors. These are receptors that signal either through G protein I, G alpha I, in which case they'll inhibit cyclic AMP production, or G alpha S, in which the S stands for stimulating cyclic AMP production. This cyclic AMP production occurs only at the plasma membrane because these receptors are only active at the plasma membrane. So remember, signal input, only GPCRs. Signal output, modulation of cyclic AMP. Where in a cell? Only plasma membrane and maybe a little bit on the endosomes. And for how long? Few seconds. Few seconds is done. So consistent with that, although 40% of all drugs that are currently marketed are targeted against GPCRs, not a single one of them, other than a hormone therapy, not a single one of them has worked for any chronic disease. Not fibrosis, not cancer, not anything that is chronic smoldering. Think about it. It's few seconds, very finite. It's only in one place in a cell. But 40% of the drugs, I'm pretty sure almost everybody in this room has used a drug that targets GPCR at some point in their life. Some may use multiple of them. Now that is how cyclic AMP is generated. But that isn't the target in cancer. Turns out we have found the only other way to activate and modulate G proteins by this family of molecules that we serendipitously stumbled upon. We named them GEMs, and I'll spend a few minutes to explain what that catchy name may mean. It means guanine nucleotide exchange modulator. So just like GPCRs, these can activate G protein alpha I, and therefore that'll inhibit the cyclic AMP, but it can also inhibit the G alpha S, the stimulatory one, which is supposed to activate cyclic AMP, but if you inhibit it, you're going to inhibit cyclic AMP as a whole. It is also unique in the sense they are cytosolic proteins, and they can trap incoming signal from any receptor on the cell surface. And by that, I really mean any. We have tested so far a variety of different families of receptors. Any growth factor receptor, Wnt receptor, G protein couple receptor, integrin receptors, TGF-beta receptors, doesn't matter. You throw anything at a cell, it'll go through that node. And it was kind of spooky in the beginning. We didn't believe it ourselves. But turns out, no matter who gets tickled on the surface, they will converge on utilizing these GEMs to eventually activate the G-alpha or inhibit the G-alphas to get a coordinated reduction of cyclic AMP. But what is key is look at the next 2 points. It can happen anywhere on the cell, any membrane. Golgi, endosomes, plasma membrane, doesn't matter. It's a cytosolic protein. It's a bomb going off inside the cell. And look at this one. It's prolonged signaling. It's a smoldering form of signaling. We see it go on for hours if it's stimulated. So this happened, and now I'm going to just summarize to you what we have found in 5 years in just one slide, is you're going to ask me, so what? You have found a way to reduce cyclic AMP, and you wanted to increase it, didn't you? Well, that's because we have also demonstrated by just surgically creating point mutations and abolishing this interface that do not let the GEM proteins to activate or inhibit the G proteins, you can immediately simply reset any disease that is chronic. So let me give you an example in the next slide. We went ahead with a couple of diseases in mind, and here's how we went about. This is, I'm just showing you diverse signals come in, it goes out, and it's gated by this little area where the small, more rigid control happens. And let's think about cyclic AMP working here. It's known that normal physiology needs oscillations within a certain range. Can't be too high, can't be too low. And that's called Goldilocks, right? So nerve myelination, synaptic plasticity, memory, mood, cardiac contraction, you name it, all of it. It's essential to make sure cyclic AMP oscillates within a certain amplitude. And then we showed that if you have high levels or low levels of, or just turn off the GEM or turn them on, you have a difference. So there are diseases which have got very high cyclic AMP. That is acute cardiac ischemia with during infarction, there's a heart attack, type 2 diabetes, schizophrenia, they all have disorder of high cyclic AMP. And of course, our GEMs are very low or switched off. If they have very low cyclic AMP, these are the diseases that are characterized by low cyclic AMP conditions, Alzheimer's, cancer invasion, fibrosis, wound healing, so on and so forth. And they have got tremendous upregulation of activity of the GEMs. And in these diseases, we have been able to put in a blocker and reverse most of them. We have been able to put back the active component and reverse diabetes here. We have, of course, not gone into schizophrenia yet or the other conditions. So having said that, let me get back to what are we innovating. So with the help of Donna over the last 7, 8 years that I've had my own lab, we have come far. So we have We have gone into recently solved the structure of how it actually binds and activates G protein. You may be thinking, why? Why do we need it? Because that's the only way to get a small molecule inhibitor to actually block that activity of all that I was talking about. Simply blocking that activity is going to be enough to immediately increase the cyclic AMP levels and get all the good effects of it. Because we know the structure, we have been able to go into the human genome and find every other possible member of this family, which there aren't very many, but we'll talk about it later. And then, because we have the structure, we have been approached by a small business in Wisconsin who has joined the cause to find the inhibitor for our target in a structure-based, informed way. And because, here's the easier part, because every time there's a cancer, doesn't matter what kind, these GAM levels are through the roof if the patient had a bad outcome. So it was a very consistent prognostic marker. High levels, very poor outcome. Low levels, patients live very long. We did it not just in solid tumor, but recently in circulating tumor cells. We generated a diagnostic-grade antibodies with Roche. We went into clinical trials, and last year, just this last year, Roche had a multi-center clinical trial on just colorectal cancer, but demonstrated that just seeing the level of expression of one of these gems, they could categorize patients into who might benefit from chemotherapy while sparing those who don't need that toxic drug. So having said that, I think I was just going to go back to this Hydra, because I started there. You see, right now, molecular medicine has reached that point where if you have a tumor, we are fairly capable of looking for the receptor to target. Breast cancer, sure, ER, PR positive, estrogen, progesterone, let's give tamoxifen. Oh, and HER2 is positive, let's give Herceptin or lapatinib. Oh, yes, glioblastoma multiforme, EGF receptor, let's block that one. Lung cancer, oh, yes, EGF receptor block it. Every cancer, we keep blocking the receptors. What happens? What happens in 3 months, 4 months, 5 months? The tumor just decides to switch its addiction. From whatever we block, it just goes and hooks onto another receptor. And then it's all the same. So let me give you an example. Beautiful work done by Rene Bernard Shaw lab demonstrated from one single patient receiving EGF receptor blockade, developed resistance, huge relapse, took the tumor, went back to the lab, found, oh, the tumor has now started signaling with c-Met receptor, made that receptor high, and started working with that because EGF receptor was blocked, right? One receptor blocked, use the other one. So think about it this way. The futility of keeping on chopping each head at a time has shown us repeatedly that it's just a matter of time. That tumor cell or other tumor cell clones within the same tumor would be more than happy to switch its addiction from heroin to cocaine and just keep flourishing after that. So instead, what we have what the results and about 7 or 8 years of work right now is culminating in is actually why even bother? Looks like these gems are going to capture anything coming at it from any receptor. Why don't we leave the receptors alone? Why don't we simply reset? If the gem is off, turn it back on, which we did it in diabetes. Because in diabetes, your muscles need to react to insulin and take up all the glucose. But it's lazy, it won't do it, fine, because the gem is off apparently. So we went in, turned it back on, it happily took up the glucose. In cancer, it's opposite. It's actually too much on, too many molecules and too much hyperactive. So we went in, created the inactive form of it, and then it could simply reset it. And the other advantage is I don't need to know what upstream pathway is driving that tumor. I don't need to know what downstream pathways are driving that tumor. So the famous line here is Donald Rumsfeld, 2002, Iraq War. There are known knowns, there are known unknowns. But in cancer, while treating a patient, there is just unknown unknowns. We have no clue when the addiction will switch from whatever I'm targeting today into which other next addiction. And so this is therefore a far superior way in our mind to modulate this program or aberration in program and kind of get at it. So with that, I will stop. I know I might have touched upon a lot of different aspects from internet to biology, but we've been fortunate to have recently been joined by 4 different groups to just take this new paradigm in signal transduction. From Jacobs School of Engineering, one of the assistant professors there dropped by my office and said, I want to mathematically model the entire paradigm and see if this holds true. And it holds true. We're about to put that out in a manuscript. And as I said, from Skaggs School of Pharmacy, we've been incredibly fortunate to have computational modeling and structure biologists joining this cause to solve the structure with us so that then we can go towards drug discovery. And of course, as I mentioned before, Roche has stepped in to take it towards biomarker, and I think it was international patent was just filed. I received a letter at home, so looks like it's moving forward. So with that, I'll take questions. I'm hoping I can answer some of your queries. Thank you.
Do you want to sit or do you want to stand? Okay, Dr. Ghosh is going to stand because she likes to stand. So could I ask anyone to come up and ask the first question, or should I ask the first?
How do you know when you know that you have an answer?
How do you know when? Here, take the microphone.
I'm sorry, you're asking how will you know?
How will you know when you know you have an answer?
Okay. Well, how will you know when you know that you'll have an answer? How will you know? Well, you mean to get something working in patients? Yeah. Okay. So as far as I said, for prognostic biomarker is going towards a diagnostic kit right now because a company came forward and can take it forward. As far as drugs, we are looking for a 1-year timeline to have very good hits. But as you may know, that there's a delay in time from experimentation to the death valley of translation and then there. Now, I must mention in that, and thank you for bringing this up, if you go to the NCI website, it's pretty sobering. It'll say that most preclinical trials never see the light of the day. Means all those wonderful drugs that work in mice never worked properly in the humans. We are very much aware that such a thing may or may not happen. There's one reason why I'm optimistic that won't happen, such a thing won't happen. And that's because this pathway wasn't about one pathway or one molecule. It had something so fundamental that it wasn't just breast cancer. It wasn't just colon cancer or prostate. You know, we first worked on colon and breast cancer, and then the entire, across the world, people came out and said every other solid tumor they worked, it looked right for, as far as its ability to tell that this patient will live versus not. Finding a drug and having it work is another layer of it. But we are doing our best. We're even making tiny probes to put it in actual living animal or human being and be able to tell, okay, that tumor looks like it has the potential to metastasize, but not yet metastasized. So this is just a small point. You know, we have gotten very good at finding metastasis. We have PET, CT, molecular imaging. It's very, very good nowadays. Resolution is increased. But to see the potential for metastasis before it has metastasized can actually help way more, because we can identify those to treat and then spare those we don't want to give that much side effects. And the way we are doing that is by engineering a probe that will only monitor the amount of signal going through that one kind of molecule, no matter which receptor on top is mutated or just high expression or whatever genetic programming went wrong. So in other words, that's another reason I have faith, because it is so unbiased. It doesn't have bias towards a tissue, doesn't have bias towards a pathway or a receptor or anybody's favorite pathway. So that's why we're hopeful. So I don't know if that answered, but it's a long-winded answer.
You have a question here?
Yes. What's the first indication that you would go after? You're talking about metastasis a lot. Is there a bias toward a RTK, a receptor tyrosine kinase, or a G protein that you're interested in? And will personalized medicine direct the choice?
Thank you. So the first part of the question is whether... did I turn it off? So the first part of the question was, how will you go about choosing first whether it is towards a receptor tyrosine kinase? So it is related to the answer I just gave. In all our experiments, we remain completely unbiased. Let me tell you how. Have you seen most of the labs, if they are going to go ahead and test a target, they always activate the pathway they study. If they're studying EGF receptor, they'll put some EGF growth factor, basically, on the cells, and then they study. If they study insulin, they put insulin. If they study TGF. We put just serum. We just keep them in serum. Serum means just like our, of course, in laboratory it is cow serum, but serum has every growth factor. Everybody's favorite pathway, everybody's favorite molecule is stimulating it. Even in that setting, we can reset every aberrant program. If A was high, B was low, after we turned it off, B became high, A became low. So it is very unbiased. Secondly, you said, how about personalizing the treatment? So that is that was actually easier for me to answer, because the studies with Roche and the other studies going on, we actually have diagnostic grade antibodies to see whose tumors need this target. And those who don't express the target, they have a 5-year life expectancy of very good, almost 100%. So we don't even need to target those who don't have this hypersignaling going on.
I can be really loud, or try. So one of this is a particularly potent pathway because every cell has this and needs it to proliferate in whatever way it's going. Which would also mean sort of systemic injection of your small molecule drug.
I know where you're going, yes. 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 So targeting a targeted therapy is a huge, huge issue. But there's something that has come up in the last 2 weeks that can actually make me answer this question without feeling completely lost. And here is it. Do you remember I showed you ATP? You know, the biggest gift to us right now for this is that every cell in your body has ATP concentrations that you wouldn't know what to do with them. Plenty. There is only one kind of tissue where in a cancer patient that has the level of ATP that it has. So just to show you, on this side, you have a normal tissue. Normally, you'll eat up 1 glucose molecule and make, but 1 mole of glucose is going to make 36 moles of ATP. Okay? Just a quick math will do it. This is a normal differentiated tissue, if you have oxygen, which we should all be breathing, we should be making 36 moles of ATP per mole of glucose. But this is how tumor tissue behaves. That is 4 moles of ATP per mole of glucose. Even though the tumor eats up 10-fold more glucose than every other cell in your body, it always has low bank account. If ATP is the currency of the cell, it distinguishes itself from rest of the cells because it has very low bank account, very low ATP. So now imagine, turns out by mathematical modeling, we have right now accomplished that, is if you sway around the cyclic AMP levels, and which is a drag on the ATP of a cell, it doesn't do much in a normal cell at all. Only in tumor cells. Now, just to tell you, I have here a picture of a gentleman on the top left corner. I better tell you who that is in case you don't know. That's Otto Warburg. Again, it's an invariant principle of cancer that every cancer is acidic pH and low energy. It's not the Donald Trump telling Hillary Clinton low energy. It just sounded like that. I'm sorry. But it is extremely low energy state, and it is acidic pH because of lactic acid. So this tissue is so different from normal that cancer was called a metabolic disease. We will take advantage of that very same thing to go ahead and change the cyclic AMP status, elevate the levels of cyclic AMP, and hope to make that big difference, because every tumor cell with high cyclic AMP, either it undergoes apoptosis or stops dividing or stops invading or all three. So it should be a viable strategy without doing much to a differentiated tissue.
I can be allowed too. Thank you for this fantastic presentation and congratulations on these groundbreaking discoveries. You mentioned that other researchers across the world are calling you and wanting to share results, compare notes. How much validation exists? As you know, validation from other groups is essential to verify and ratify what we're learning. Can you talk a little bit about that, about how the rest of the world is coming on board with your research, and what are you learning about those interactions?
Yes, actually, very, very important point, and I should tell you that I have had a lab only for 8 years. And one of the advices of my mentors that I probably didn't take is don't start your career with something where you come out swinging a bat. So what happened in the beginning of my career, I was slapped around from growth factor people saying it's impossible. I still have reviews from grants that I wrote where they said this is impossible. I have, same thing happened with G protein side. They said it's impossible. So now let me fast forward to 7 years, because last year we came a full circle. So Brian Kobilka, Nobel laureate 2012, and Lefkowitz, who were the latest G protein coupled receptor Nobel laureates. I told you that it has been 8 times that this thing has gotten recognition, because every one of us use those drugs. It's the 40% of drugs. So he called me over to present in a crowd who doesn't care about what we do. They only care about the GPCR part of it. It was a wonderful experience for me. They all came up, congratulated us for just new biology that across the disease. The diseases that GPCRs can't do nothing about, like fibrosis, like type 2 diabetes, like cancer, right? They came and congratulated, and right away they said, we'll believe it when you have a structure for it. So that's what last year happened. Thankfully, somebody here in Skaggs approached and helped us get there. I couldn't have done it myself, of course. And then somebody else came and said, we'll believe it if you can demonstrate some other parts of it, which we did this year. And the reviews came back, of course it had to be this way. You have been telling this for the past 7 years. So you can get it both ways. And then finally, I must tell you that it is good and bad. So I had the bad side of it some years ago. Right now, the flavor of the reviews are, you have monopolized it. Which is good. No, which is good. Because in the last 2 cycles at NIH, I made sure I'm not the lead PI. There are other fields who joined us in this cause, not me. So I was at the back. I stepped behind. There are 2 other PIs in front from totally different fields. One for mathematical modeling from an engineer. One from a molecular imaging person in U, Michigan, who joined our cause because he's the best at doing in vivo imaging. That's the person with whom we are trying to develop these tiny probes to measure the potential of metastasis for the first time. Not metastasis, but the potential. And then finally, the structure folks. So those are the 3 things that went out. It is, I've been told that you cannot hammer in and say, I told you so, so you have to believe it. It doesn't work that way. So I was told that you have to take people along with you. If they are non-believers, they will believe when they get involved, see, test, and figure it out. So when I started, there were exactly 5 manuscripts on this first protein. Right now, it's 116 as of today. When I started, we only did colon and breast cancer. Right now, its importance has been across every solid tumor. I didn't put up that slide, but now I'm thinking I should have. Anything you can think of, from glioblastoma multiforme to prostate cancer. And then we are lagging behind in getting the drugs, but we've been busy with the biology. Now that we have more critical mass, and critical mass was important for not just being a one-person, one-lab finding, but going ahead across the fields, testing it with math, testing it with physics, testing it with imaging, testing it with all possible avenues. That's what I'm hoping will bring believability.
Quick question on the issue of collaboration and,
Yes.
and all these different fields and different scientists around the globe. Can you tell us a little bit more about the collaborations here at UC San Diego and in San Diego a little bit more broadly? What about colleagues at Moore's Cancer Center? What about the Mesa here, Salk, Sanford, Berlin, etc.?
Thank you. So it's about collaboration. Most of my current existing collaborations are with SCAG School of Pharmacy and Pharmaceutical Sciences, Jacobs School of Engineering, Mechanical and Aerospace Engineering, Radiology Department, but not local. That is in Michigan, Department of Radiology. And as far as drug development, we have been with Bellbrook Labs right now, but I will be going to Conrad Prebys, and then at Moore's Cancer Center, I know Scott Lippman has put every resource possible, the medicinal chemist, to finally make it translate into the usable compound as needed. As far as the other Moore's Cancer faculties, I have sporadic collaborations, but not on this topic per se. I think a lot of the last 7 years has gone into the biology. Translating takes a little bit of more momentum where others believe what we see and test and then move on. But I think we'll have more soon, hopefully. Yes. Oh, sorry.
Yeah, one more. It's a bit scary
Oops.
because if you can do this for the cancers, any tumor cell of this type, you can do it also with any living cell of any type at all. Is that scary?
So if I understand the question correctly, if you can do this to any cancer cell, you could also flip it in the reverse direction for any, like anything else in medicine, that is a possibility to design an agonist of this pathway. to make it better. Now, there is a condition where we want it better. You would want it better too, I'm sure, which is type 2 diabetes.
I'm too old.
Turns out, type 2 diabetes, these are not firing. So when we went in and made them on, like perpetually on, there was no more insulin resistance. But then, targeting that needs to be done carefully because otherwise we're going to predispose the whole person to multiple sites of cancer. So you're absolutely right. But we're not looking for agonists right now. We're going at blockers.
But somebody will.
May, yes.
Oh, I was just going to ask, you're doing the downregulation of the small molecule. What are your thoughts on upregulating?
Yeah, so right now we have gone forward with peptidomimetics, which is small pieces of protein that have the active component, and we have only looked at it in the context of type 2 diabetes. I have not tested it any other condition because the other two conditions in which we have published there's a need to block is two conditions that don't have any multi-receptor targeting therapy, which is fibrosis. There is no current treatment known to reverse fibrosis anywhere in the body. Think about it. There are about millions of receptors, all of whom can promote fibrosis. Again, it's a multi-receptor process. Again, it's a chronic process. Again, it has no current therapy. And the other was cancer progression and invasion. So though we are designing blockers with those two diseases in mind. If we were to design something to make it better, it would be diabetes in mind. But I did show you other conditions like Alzheimer's and schizophrenia. I have not gone ahead with those specific conditions in mind because you have to think about blood-brain barrier and other things if you want to get something into the brain. So it's a little bit more complicated targeting.
One last question. Are there any, or have you looked to the existing libraries of compounds out there to see if any of them would have any sort of the expected phenotypic effect when administered in any kind of models that you would see consistent with an inhibitor of these GEMs?
So there are no current molecules that are fitting into that structure. So we don't have we have a global G protein blocker, but nobody here would want that.
Sure.
Because it'll make your blood pressure shoot and everything else go wrong because you're going to block GPCR signaling, and we don't want that. Now, something I didn't mention, you can actually target this and not affect a single GPCR in your body. Because turns out, because of the structure we have solved, we now know the place where the GPCRs come and activate the G protein is completely on the opposite side of wherever these molecules activate the G protein. So it's a totally legitimate, plausible, feasible target, like in a structure sense. And so the question is how we are going to take the hits we are going to get, the small molecule hits, and move forward. Ruben, one of the things you asked is repurposing something already there. But we don't have anything right now that kind of fits that pocket, the drug pocket.
Thank you. Any other questions?
Yes.
Is there a possibility that your methodology and approach might in the future apply to non-solid tumors?
So the question is whether this would apply to non-solid tumors. The answer is yes. Now my question there would be non-solid tumors typically have homogeneity to some extent, like it comes from one clone, stays clonal. Currently, though, some thoughts are changing on that some of those liquid tumors we thought are monoclonal, like all coming from that one cell and looks alike, may not be so. And even there, when it's targeted, maybe it'll develop more clones during treatment. So one of the groups in Germany did approach us to take it into leukemia, lymphomas, and we have been trying to help them out to see if they can take it there. I myself am not a hematologist, but if I were, it would have been a lot easier working with liquid tumors. Blood is easy to get, right? Tumor tissue is just a little harder. But solid tumor has this heterogeneity that has been so difficult to deal with, such a challenge, that we took that as our first, we went into that and we've kind of stayed in that for a while. And also the receptor blockade as a treatment protocol is more for solid tumor. Liquid tumors, as you may know, Gleevec, it's a kinase blocker. Kinase is inside the cell. With solid tumors, they always are blocking receptors, always blocking a receptor on the surface. So we went forward with that just because of the biology. Thank you.
That was a great talk.
Thank you.
I always like to hear your talks. Is there any data, I'm asking this because we have such data, that would show how long ago the picture and the pathways exist in the body? If you could get really old cohorts that you had blood out from for a very long time, it seemed like that might be very helpful in knowing when and how this marker evolved. And if you have a lot of other behaviors, smoking being the obvious one, that would also be helpful. So I think it might be fun to see whether or not we could put a little project together to do that with our aging cohort that's as old as I am or older, and it would be maybe useful to know when things begin to change direction.
Absolutely. You've hit on the one of the things that actually one of the projects, two projects in the lab that are absolutely fascinating but has nothing got to do with the grants that feed my lab. I hope they never find out I use the money for this. One is male infertility, and the other is aging. Male infertility, turns out if you just look at the 500 gene signature of infertile men, all published out there, we always go with big data first to see if we are in the right direction. But again, we're looking where the light is, sort of street light effect still, but just so you know. So in sperms, turns out you need that to be activated, the gems to be activated so that it can actually capacitate and fertilize an egg. And if you turn it off, then you end up getting premature acrosomal reaction, basically no fertility. The reason I bring that up, because the second one is aging, and you mentioned that. These changes must be happening over time in a pre-neoplastic state. And we have recently discovered some of the molecular pathways that are showing us that. For example, in a recently accepted work, should come out in a month or two, we have found out that what do these things do in a normal cell? In a normal cell, they hang around near the top of a cell, holding the junctions of the cells together and maintaining order. And then, lo and behold, if you constantly barrage the epithelium, all cancers arise in epithelium, right? So if you barrage the epithelium with constant stress, energetic stress, caustic stress, reactive oxygen, constant stress, the junctions fall apart. Slowly they become loose. And then we could demonstrate that a specific event happens, lets those move away from the junctions fall flat, cells become dysplastic, and they move around and they start growing into tumors. We also demonstrated, how many of you here take metformin? So we also demonstrated a simple anti-diabetic drug like metformin would make sure that those are kept right where they belong to maintain orderliness in the normal epithelium. And that is actually one of the tumor suppressors that not well known as to how epithelium is protected, but here. So I would love to look at pre-cancer, the duration it takes to see some of these earliest changes from normal, and then the dysplastic stage, if it comes off from where it should be in normal, and then it levels sort up, and then how it progresses as the tumors develop. So maybe we should talk so that I can follow up on that, but great question, thank you.
Anyone else? Well, let's thank Pradipta, please, for a fantastic presentation.
Thank you very much.
And we invite all of you to please stick around. We have food outside for everyone, and we'll have an opportunity for you to interact with Pradipta and ourselves.
Thank you very much for coming, and look forward to seeing you here next month, the third Tuesday of the month. Tell your friends about this series, and thank you for being here.